As we sort of put the final touches here, a couple of things. Remember to stay for the business meeting. We'll make it worth your while. And then the reception at 5:30 at the, what's it called?
Meeting court.
And which is?
Apparently, Shirley says 1 block outside the building that way on International Drive.
Okay.
That way.
That way.
Sorry. Go to the war.
Not doing whatever that way is. Excellent. So are we ready to work it over? Very definitely. So continuing with the award section here, the Young Investigator Award, what I refer to as the Young Pump Award, goes to Lane Baker from Indiana University. I will let Dick Crooks do the introduction, and then John will present the question.
Okay, thanks everybody for sticking around for the second part of the symposium. It's my pleasure to introduce News is something else completely, and I wouldn't do it with Lane. I wouldn't do it here. Introduce Lane Baker. Lane started off as an undergraduate in Missouri, Southwest Missouri State, something along those lines?
Exactly along those lines.
All right, Southwest Missouri State University. And then he joined my group for his PhD when I was at Texas A&M University, and worked on a number of different projects, including some synthesis, interestingly enough, that was quite successful, in addition to electrochemistry and surface chemistry. And then he came in one day and told me that he was going to take a postdoc at Naval Research Lab, and I was really surprised in a way. Not that he was going to NRL, but that he was kind of the group he was going to work with. There were some parts of the puzzle that didn't quite fit together, but off he went. It turned out there was a postdoc in the group. At the time, Lane was a graduate student who took a job in the Washington D.C. area, and it turned out she was the reason Lane was in Washington. That was successful. He married her. They have offspring. And so he completed his postdoc there, very prestigious NRC postdoc.
Is this being webcast?
Well, we unplugged it all. and then did a second postdoc with Chuck Martin at the University of Florida. And that was also another extremely successful postdoctoral stint. And it was during that time that he became interested in nanopores. And he's continued his own career at Indiana University studying nanopores, both as scanning probe tips and also in an interest in the pores themselves in, for example, membranes and things like that. So, and then I think that's what Lane's going to tell us about after Jana steps up here and gives the award.
Hi, Lane. Are you going to come down or am I coming down?
I'll come down.
So this is the Society for
We can't hear you.
Society for Electrochemical Chemistry presents the Young Investigator Award for 2012 to Lane Baker.
Okay, so thank you very much. And today I'd like to tell you about some work that my group's been doing on measuring gaps in spaces with ion conductance microscopy. I've shown my entire group here because it's really a group effort. And I'll try to point out at the end some of the people who made some of the measurements that I'll show you. So by way of an outline, I'm going to start by giving you a little bit of motivation for why we're interested in this topic. Then I'll tell you about this technique of scanning ion conductance microscopy and the measurements that we've made recently with this technique on single pores and on multiple pores in a single membrane. And I'll wrap up by telling you about how we're going to take this, or where we're going to take this in the future, and some conclusions. So, kind of an overall interest in our group is the idea of ion transport in tissues. And so this turns out to be relatively complex, and it's difficult to study at small scales. And what we'd really like to do is be able to study ion transport at subcellular levels, so levels less than a micron, hundreds of nanometers or smaller. And there are many different mechanisms at play here. There can be molecular mechanisms, intracellular, extracellular features or factors, but these processes are critical in a wide range of processes and diseases, pathological states. So what you can see here are two images, and these are images of stained histology sections of cuboidal epithelium in the kidney tubules. You can see that this layer of cells lines up, and it forms a nice barrier to transport of ions across this tissue, into and out of this tissue. And so ultimately, this is what we're interested in studying, are these kinds of processes. What I'm going to tell you about today are polymer membranes. And we've chosen polymer membranes because they provide a very well-defined membrane, very well-defined regions of heterogeneous or homogeneous transport through these pores. This is a cross-section of a polyimide membrane. It's about between 12.5 and 25 microns thick. And what we'd like to do is be able to examine the transport of ions through these pores. These membranes are interesting in and of themselves for a number of reasons, but then they also provide a well-defined model for future systems related to biology. So the way we're going to study this is using an electrochemical scanning microscope. And so I've put two examples of electrochemical scanning microscopes here. I've put a scanning electrochemical microscope, SECM, and a scanning ion conductance microscope, SICM. So most people are probably familiar with the scanning electrochemical microscope. Bard and others made this very popular, where a small electrode is approached to a surface, and the electrochemical processes either at this probe tip or between this probe tip and the surface of some redox mediator are investigated as a function of position. Now, kind of the ugly sister or ugly cousin of scanning electrochemical microscopy is SICM, at least in the electrochemist viewpoint. The reason why is there's no Faradaic electron transfer here. We're just looking at the flow of ions between this electrode and another electrode present in the bath solution. These ions flow through this resistance, and we use the changes in resistance to position the pipette. The feedback mechanism for SICM turns out to be very similar to the feedback mechanism for negative feedback scanning electrochemical microscopy. So in the end, both of these techniques are surface sensitive. The SECM relies on a Faraday current. SICM measures differences in conductance right at the tip of the pipette. Now the big advantage, though, or why we're really interested in SICM is because of resolution. And probe fabrication with SICM is very simple. You don't have to spend inordinate efforts to try to get very small tips that have the proper geometries. If you try to get the same kinds of tips for SECM, you have to go to really heroic efforts. But we can generate lots of tips in a very short amount of time that give us high resolution, both spatial and in terms of sensitivity. So everything that I'll tell you about today is about the ugly sister. I hope that doesn't mean I'm attracted. Well, this is webcast, and Dick's already involved my wife, so I'm not going to go any farther with any of that. Okay. These are the tips that we use. So they're really easy to use. This is the same kind of tip that you use when you do a patch clamp experiment, except instead of trying to get a tip that's microns in diameter, you try to get a tip that's nanometers, tens of nanometers, hundreds of nanometers. There's an optical image on the left here of a pipette that we've pulled. And then there's 3 scanning electron micrographs of pipettes. There's 1 scanning electron micrograph of 3 pipettes shown here. In this example, one of my students pulled this down to a very fine taper so you can see the draw of the pipette. Now, in the final image, you'll see a scanning transmission electron micrograph. And so this is an image of electrons actually going through the tip or the business end of the pipette. And what you can see is it's relatively easy to get, you can see that we can get pipettes with diameters on the order of 100 nanometers or smaller. And it turns out to be relatively easy to get these kinds of pipettes. Now, if we look at the end-on views of these pipettes. You can see here's a single barrel pipette, here's a double barrel pipette. I can't tell if the contrast is bad or if it's just this LED light that's blinding me. But we can routinely get these small diameters, and what's most important about it is that we can get appropriate RG values. So we can get appropriate ratios of the active element of the electrode to the outer diameter very easily, and that lets you image very close at high resolution. Now, there are other ways to increase the chemical information if you're interested in doing electrochemical measurements, using theta pipettes or chemically modifying the outside of the surface. So in this pipette, there's a barrel here and there's a second barrel here, and you can use one position or one of the barrels to control the position of the pipette, and you can collect an additional signal through that other barrel. Now, we've explored chemical modification at some levels, and these are with these dual probe electrodes where half of the electrode's covered with a conductive layer. In this case, it's gold. And if you're interested in this combined approach of SECM, SICM, one of my students will be talking about it Wednesday morning at 8:30 or 8:20. So the other thing about this technique that we do, or that's different from typical SECM experiments, is that we use a modulated feedback. And so Bard developed this for SECM, but the SICM community picked it up. And so what we do is we actually vibrate the tip in the Z direction in space. What that does is when the tip gets very close to the surface, this modulation produces a modulated resistance between the tip and the surface, and we can use that to control the feedback loop then. And so by looking at the peak-to-peak change in this current, we can get a probe surface approach curve that looks like this, where if we're far away, there's no change in the ion current as a function of position. And then as we get closer, the modulated peak-to-peak amplitude picks up. And so that lets us then select a scan distance. So if our probes, this is the radius of the probe, if our probe radius is on the order of 45 nanometers, we can then select a scan distance relative to that radius and use that current value as a set point to control the position of the probe relative to a surface of interest. This is important because it's surface sensitive, so any DC drift out here in solution doesn't affect the measurement. And it's also very low background. And so it's very advantageous for the measurements that we're interested in making. Now, on the experiments that we've done, I'm going to tell you about two situations, a situation where we measured single pores and a situation where we measured a membrane that has multiple pores in it and finally many pores. So a typical SECM or SICM experiment, we have two electrodes, a pipette electrode that's inserted into the pipette and a reference electrode that's outside in the bath solution. The pipette's filled with an electrolyte, a conductive electrolyte. The outside medium is filled with a conductive electrolyte too. Now what we're going to do though is add in this case, an additional electrode, right? So a third electrode that we're going to call the working electrode in this case. It's not a working electrode in a true electrochemical sense. But we call this a working electrode because we varied the potential on this electrode, right? So the pipette electrode is held at a constant potential, usually 100 millivolts relative to the ground. And then in certain cases, we'll vary the potential that's applied to the working electrode. They're both tied to the same reference electrode. And what we'll watch is how this potential difference affects what we observe at the pipette. The system that we're looking at here is a perfusion cell. So there's a membrane that has one pore in it. And that one membrane with one pore in this first case is separates two halves of a conductivity cell. The electrolyte, we've got homogeneous electrolyte concentration, so it's 100 millimolar KCl in the pipette on top and below. And these two electrodes are in the top half of the chamber, and the working electrodes then in the bottom half of the chamber. So here's the membrane that we're actually looking at. We use a very low-tech method of isolating this pore with an epoxy under a microscope. And so you can see here in an optical micrograph, you can see this border here shows everywhere inside of here is not covered with epoxy, everywhere outside is covered with epoxy. And so just this small region of membrane here is what's actually exposed. And we can go in after we've done experiments and we can pick out where this pore is at with an electron microscope, and we can zoom in and get the pore dimensions of the pore exactly. And that helps us in terms of trying to understand if the theory or the models that we're developing are actually correct. Okay, there are two ways to make these kinds of measurements then. The first way is that we're going to take the tip and we're going to raster it across the surface in an imaging fashion. When we do that, we hold the pipette electrode at a constant potential, and we hold the working electrode at a constant potential. So the only thing we're looking at then are differences in the spatial conductance profile. The other thing that we can do is if we find a spot, for instance, over a single pore, where we see changes in the conductance, we can park the tip at the pore and scan the working electrode potential. And when we do that, we turn the feedback off so that the position of the probe doesn't move up or down. So the probe's locked in position relative to the pore. And we're just looking at the influence of these two electrodes on each other relative to the reference electrode. When we raster the surface, we obviously have to have the feedback on that maintains a constant tip sample separation. Okay, so if we have this rastered pipette situation with the 3 electrode setup, we can record topographic and ion current images at the same time. So what we're measuring then are the deflection of the piezo and the Z, as well as the DC component of the ion current. And so here in the topography, there's a pore right here. It shows up in the image here. This is a micron for comparison. If we hold this working electrode, so these are held at constant potentials. If we hold the working electrode at ground and we don't see any change in the ion current or the conductance at that pore. Right? If, however, we change the potential of this working electrode, then we notice a change in the potential that's observed at the pipette, or the current that's observed at the pipette in this case. And so if, for instance, we hold this potential at minus 300 millivolts, we notice there's an increase in the current that we measure. And if we hold the pipette electrode, or the working electrode at 300 millivolts, there's a decrease in the current measured at the pipette. So we can also look at this if we're right over a pore or if we're spatially far away from a pore. And so in this case, we have a pipette that we've positioned approximately, well, in this case, it's about 5 microns away from the center of a pore. We've locked the position in so the feedback's off, and now we're going to scan the potential on this working electrode. And what you can see is there's some change in the current voltage response, but there's not very much. The pipette's physically separated about 170 nanometers from the membrane in this case. If we take this, though, and we now park the tip right over the pore and we scan that electrode in the same way, now we get a big change in the slope of the current voltage curve. And the other thing that you'll notice is this is backwards of what you'd expect from a typical current voltage curve, but I'll explain that in a moment. So it looks like, based on where the pipette is in space relative to this conductance pathway, we can measure changes in the current that are measured through the pipette. Okay, so how does this work? Well, my students take Gary Heafey's electronics class, and he's in the audience, so everything looks like an electronic circuit to them. And so if you take this and you put it in a circuit, in a simple circuit simulator, we can have a pipette electrode, a reference electrode, a working electrode. Then we can have an access resistance. This is a resistance that's defined by the gap between this pipette and the membrane. And then we can have the resistance of the pore. All right. So that's just a simple example. We can also obviously have capacitances involved. But there's 2 situations. If the pipette is positioned far away from the pore, then the access resistance and the pipette are in this top half of the current loop. And the pore is isolated. The pore is isolated in the bottom half. But if we move the pipette over the pore, then the position of this access resistance changes. And so this works kind of like a voltage divider, where the top half and the bottom half now both have to go through the access resistance to see the reference electrode. So if we actually put these values into the circuit simulator, we can calculate the potential at this point, and that can give us an idea of why we see the current changes that we see. So we're using realistic or measured values for the pipette resistance, the access resistance, and the pore resistance. And if we do that with a constant potential applied to the pipette electrode and then vary the potential between the reference electrode and the working electrode, this electrode in the bottom half of the chamber. If we have, we start with the case of 0, where we have no potential applied to the working electrode, we see a very small change at this point, or this junction, when the pipette's directly over the pore. So we can use this for our baseline value. If we add, or if we apply 500 millivolts to this electrode on the bottom, then we see that the potential at this point goes up to about 5 millivolts, and the current through that loop goes down by about, in this case, around 50 millivolts. If, however, we change the potential on this electrode to minus 500 millivolts, then we see the opposite effect, where the potential here in solution is lowered and the current through the pipette's raised. And so this would explain then the phenomenon that we see. The important thing to remember here is that the working electrode, the potential we apply on the working electrode, and the current through the pipette then are inversely related. So if the potential on the working electrode goes up, then the current through the pipette goes down. So if we look at our data, or our actual data shown here in one of these circuit diagrams, and we compare it to what you would calculate with this circuit model, we have very good agreement in terms of the overall magnitudes and qualitatively the shape and position of the pores, of the IB curves. Now, the other thing that we can do with this is kind of interesting is we can actually, because the diameter of the pipette is less than the actual diameter of the pore, we can advance the pipette into the pore. And so here we can take a measurement at 600 nanometers away from the surface as shown in black. But as we steadily increase or advance the position of the pipette into this cavity, we can watch the contribution of the access resistance to this process change. And so here we see an obvious difference once we're 2 microns into the pore. We're relatively deep in the pore. This has a much larger effect on the observed pipette current that we measure. Okay, so that's all well and good, but measuring a system that has 1 pore in it could be cheating. It's kind of cheating because there's only one place for the current to go, right? The current has to go through the pore and it has to go through the pipette. And that's not so interesting if you start thinking about a real system. So we took the system that we had under study and we went from a single cylindrical pore to a pore that has to a membrane that has multiple pores in it, and those pores are shaped as cones, all right? So they're conical pores. Now we have to add an additional electrode here, but it's just to take the current. It's still referenced in the same fashion. And so the idea now is if we have 2 different pores in a membrane, and those pores are shaped differently, and we park the tip over one of these pores, can we measure a difference in the conductance of this pore versus the conductance of the other pore translated through this change in potential at the pipette electrode? And so here's an optical and electron micrograph of a membrane that we've prepared. There's 6 pores that are isolated here. And the way that we've made this is through an asymmetric track etching process. So each shape has a different tip geometry, a different base geometry, and it has a different angle, a different cone angle. And so that means then that each of these pores is relatively unique in terms of its shape, and that gives it a unique conductance. And so we can go back in through all of these pores after the fact with an electron microscope, and we can find, well, we should be able to find all of these pores. We couldn't find one of the tip diameters, but this is actually a monumental task. I told the student to, just go find them. It's easy. And I was really happy that they came back with 5 because I thought they'd come back with none. But they came back with 5 out of 6. And for the base diameters, they're a little bit larger, so they're easier to find. And so we came back with all the geometries on that. But what we can see is that the base diameters range from about 650 to 700 nanometers, and then the tip diameters, there's a larger spread in terms of the overall size. They range from 700 to 150 nanometers in diameter. And there's two pores that are positioned very closely here, and that's what we can look at first. But I want to talk about just briefly the current voltage response of these pores because they're asymmetric, all right? So these subtle changes then in the pore geometry influence the conductance of these pores. And if the pore's charged and the pore's asymmetric, then the pores typically rectify, or under the right conditions, under the conditions we're studying here, they rectify. And so rectified in this case means that the current voltage curve is nonlinear. It passes a higher current, or it passes higher current at a corresponding negative potential in this case than the positive potential. And we can represent that then as the ion current rectification ratio, the ICR ratio, shown here, just the absolute value of the ratio of these two currents at a given potential. This is a really interesting phenomenon, and there are talks just about this concept in and of itself. So I don't want to go into that explanation very much here. Dick, your pointer here is killing me. But we can take one of these membranes that has a single conical pore in it, to start with, and we can look at the shape of the current voltage curve we get in that plot. And so here, if we have a pipette that's over a conical pore, and this is the arrangement of the pipette to the pore, if we're away from the pore, we get this black trace shown here. If we move the pipette over the pore, we get the red trace shown here. All right. Now, just so that we can compare this to what's published in the literature, where you have a true current voltage quadrant, we can reference the current that we measure at the pipette to the current at zero applied potential at the working electrode. And if we do that, we get a curve that looks much more familiar and makes me feel much better when we talk about the data, that looks like the current voltage curve you might get through one of these pores. Now, if we take this same pore that's measured, and we look at the current voltage plot that we measure with ion conductance microscopy, if we measure that exact same pore in a bulk measurement, where things are much more well-defined and understood, we just have macroscopic electrodes sandwiched between the membrane sandwiched between a U-tube, two halves of a U-tube cell, and you'll get the current voltage plot shown here in black. And so you see the ICR ratio is very similar, and qualitatively the curves are also very similar. So this gave us some confidence that we were actually measuring or transducing what was going on at this pore with the pipette and the electrode configuration we have. So this is just a formal definition of this delta ICM here, the pipette current reference to the pipette current at zero applied potential. We can plug this all into the same system where we've added another reference, another resistance here that is accounts for the other pores in the system. And if we do that, we can solve this with a series of known and unknown values, and we can get a relationship that says that this delta ICM that I plotted reference to zero relative to the true pore resistance should be related to the access resistance and the pipette resistance as shown here. And so the access resistance, to remind you, is this small gap here. The pipette resistance is the ionic conductance, the resistance to ionic conductance of this pipette. So again, if we do that, we can look then at this value in a typical plot that you might look at in these kinds of experiments. Now, so what's shown here are these two pores that were very close to each other. And so these pores turned out to be more cylindrical or less conical than the other pores in the membrane. And so they don't rectify very much. But what you can see is if you take the tip, they're separated by a little over a micron. And if you take the tip, if we first image at a constant working electrode potential, we can get a plot that shows us where the pores are. We can then take the tip and position it over different places in the membrane and measure the current voltage response. And you can see that if we measure at position 2 and position 4, which are directly over the centers of these pores, we see a much larger change in the current voltage plot. If we measure out at the outskirts of these pores, we see some change in slope, but it's not nearly as drastic as it is when we're parked directly over the tip. Okay, so it's definitely harder to measure when there are more pores in the membrane, but it's not impossible. And so what we can do is we can take the tip and go around each of these positions after we image them and find them. We can park the tip at the same position, turn off the feedback, and measure the current voltage curve over each of the pores. Now, if we do that, we see the current voltage plots shown here for each of these individual pores. And so each pore has a different slope. Each pore has a different rectification or no rectification, and that says that each pore has an individual or a unique conductance. But what we're really interested in here is if we are actually representing the conductance of this membrane overall in a quantitative fashion. So if we take these 6 conductances, or the values for these 6, and we add them together, you get the plot that's shown in red. So the plot that's shown in red is all measured with a pipette. It's the sum of the 6 current voltage plots. If we measure all the pores in the membrane in one shot in a U-tube cell, you get the current voltage curve shown by the black dots. Okay, and so what that says then is this measurement does reflect the current measurement that you would get out of a typical U-tube setup. Further, if you look at the equation that we developed, the change in the ICM here relative to the change the change in the ion conductance microscope current relative to the change in the pore current should be related to each other through this change through the access resistance and the pipette resistance. If we plug in values that are realistic for this system of an access resistance and a pipette resistance of 1 megohm and 90 megohms, you can see that these two values on the y-ordinates are very close. The y-axis in this case is about 1% of the y-axis in that case, and that agrees very well with these numbers. So we think we have a chance then of trying to measure these kinds of things with some quantitative effect. The other thing that this technique does that's kind of interesting is that if we have two pores that have different conductances, you can see things that are different relative to imaging. 5 minutes, okay. Relative to an imaging condition or this voltage scanning condition. And so, for instance, if we hold the electrode in the bottom half of the U-tube cell or the conductance cell at negative 500 millivolts, and we image pore 4 and 5, we can see the image of the pore 4 and pore 5 shown here. And that's reflected, that image reflects the conductance of the pore at this state. If, however, we change the potential to positive 500 millivolts, you only see the pore at position 4 show up. The pore at position 5 doesn't show up. And that's because when we're imaging over here at positive 500 millivolts, there's no conductance through the pore here. All right? So we're able to probe then this nonlinear current voltage curve by using the position of the pipette in this voltage scanning experiment. And you can see here at position D, if we're away from the center of the pore, we see relatively no change in the current voltage response. So we think we have a way then to measure these kinds of conductances, but it's not really perfect for a biological system. The reason why is because we don't want to apply 500 millivolts across a biological interface and expect it to actually retain any of its properties. And so because this looks like it's a change in the potential, we need to think about ways to measure with highly conductive samples or highly conductive membranes at low applied potentials. And so what we've started trying to do in the last month or so is to use a double barrel pipette where you have this theta pipette and one electrode is hooked up to essentially an EMF meter as opposed to measuring the current directly. So here we can probe the solution potential. This is one of these double barrel pipettes. If you can see this diameter, the inner diameter is roughly 50 nanometers in each pore. This is a porous membrane with 500 nanometer diameter pores. There's 8,000 pores in the membrane, so we went from 6 to 8,000. And the electrolyte's the same as in the other case. The important thing is the membrane resistance. So this membrane resistance is the same resistance that you would expect to measure across, for instance, a layer of epithelial cells. And so these numbers all match up well with trying to measure actual biological tissues, samples. If we try to make this measurement measuring the ion current instead of the potential, all right, if we look at 60 millivolts, from 0 to 60 millivolts, this is the range that we'd like to be able to work in. If you look at the ion current measurement, you can't see very much change if you look at the straight current. But if you look at the second barrel, which is measuring the potential, you can see a big change. Now, there's gain on this, but it's very easy to distinguish where the pores are relative to each other with this potential probe. And just because the color scales here are sometimes tweaked, we looked at this by comparing these two signals, the EMF signal versus the direct current measurement on a relative noise scale. And you can see with the EMF scale, we get about a 5 times enhancement in signal to noise ratio. This operates at very low resistance membranes with low potentials, and so we're excited about where this might take us for measuring biological samples. Today, I hope that I've showed you these things, that we can image and measure conductance properties in a non-contact manner at very small scales. We can image individual conductances, and that we can also use the aggregate of these to reflect bulk measurements. And I gave you an idea of where we're going to take this in the future. And I'd like to thank everyone in my group. This is my group at IU, especially Chao Chin Chen and Yizo took the majority of the data. If you really want to understand this, they're giving talks Thursday morning about it. and they're very well spoken. I'd really like to thank the mechanical instrument services and the electronic instrument services because they built a lot of the hardware, the electronics hardware that we use in our measurement. And especially I'd like to thank Bob Ensman. So some of you might not know Mr. Ensman. Mark's jumping up and down. But Mr. Ensman is retired from IU, but he still comes around and visits us relatively frequently. And he was nice enough to actually sponsor this award when he was the chair or the founder of Cypress Instruments. And so he's retired, and they don't sponsor it any longer. But he's a really a great asset to the electrochemical community, I think. And with that, I think funding, and I'd be happy to answer any questions. So one of them is less conical, but then the other thing is the charge could be different on each of the pores, and that would change your rectification too.
The interior.
Right. Yeah. And so physically, what we observe is that one of the pores rectifies less. One of them rectifies much more. This is really for everybody who uses polymer pores. When you start making them, sometimes they rectify, sometimes they don't rectify, sometimes they rectify a lot, sometimes they don't. But the common thing is if you have a single pore, you can just keep tweaking it until you get the conductance that you want. But in this way, we can actually probe and see what the pores, how that conductance evolves as a function of the etching process.
So do you have SEMs of these?
Yeah.
Can you see differences
Yeah.
in the pores?
Yeah, absolutely. Yeah. Yeah. So everything agrees in terms of the conductance that we measure and the geometry of the pores. This is an extreme rectifier, and it may be that this region of the membrane has higher charge than another region in the membrane.
Henry, I was curious, for biological cells, you worry about the word DC bias. Well, how about just doing SCCO, an operator to a metal probe, an operator that is AC, low voltage AC conductivity? You're still getting conductivity. You're getting that information.
Right. Yeah, but I don't know if we'll ever get a probe that is very high resolution doing that. Or the thing is, I don't want to spend all my time trying to make very small, nice electrodes, I guess. I like these pipettes because they're really fast, right? And so they're easy to use. We are looking at combining the two together, actually, to do that with a metal coating on the side of a pipette. But this is kind of like your AC stuff, right?
Yeah, we image membrane.
Yeah.
Pores similar.
Yeah.
AC image.
Right. Yeah.
In terms of that difference in rectification you have seen, have you ever tried on decorating in the surface with some different materials where you can see more of that rectification along, especially the experiment you did with the hide?
Oh, to go down into the pore and see if we can see what's rectified and not? No. Part of the problem with that is if you want to pour the well, yeah, I guess we could actually do that. Yeah, we haven't thought about doing that, but yeah, that's an interesting idea.